afternoon, everybody. I was very lucky to be the first person out of my cohort to talk, so I have the special privilege of extending the welcome to every single one of you. So really, I really want to welcome you here. Good afternoon to all the friends, family, mentors, and supporters, both here physically and over Zoom. It really meant a lot to all of us that you could be behind us throughout this entire project. So my name personally is Patrick B. If I could figure out how this mouse works, I will go to the next slide. There we go. My name is Patrick B. I had the special privilege of working in the Dr. Zhang / Ellenbogen lab for the past 8 weeks. And I was working on a project specifically geared towards the delivery of gene therapy towards breast cancer cells using nanoparticles. I thought I'd like to give a little bit of background to what our project focus was and the motivations for it. I think we're all somewhat familiar with what cancer is, but to delve a little bit deeper, cancers have a few characteristic properties to them, one of which is that they grow very quickly, they proliferate very quickly, and the other part is they rapidly accumulate a lot of genetic mutations. So normally our cells, whenever they mutate, have repair mechanisms that will repair those mutations so our cells can function normally. Cancer cells have a property where those accumulate and are left unrepaired. Some advantages this gives to cancer, unfortunately, is that it makes them survive a lot better. Maybe not individual cells, but as a group, they can accumulate many mutations that allow them to survive and proliferate better than what our normal cells would be able to do. So to combat this, we've developed multiple therapies to treat cancer patients and cancer cells, but it seems that the ability for cancers to survive and develop resistances through these mutations is outpacing the therapies that we're able to develop so far. A couple of the things that we're also able to do is excise tumors physically using surgery. This is a common method of removing tumors, but unfortunately, through many tumor removal processes, it's hard to remove everything, and some microscopic cancer cell clusters are often missed as a result, and these can regrow, and honestly, those that are left have these mutations that allow them to be even more resistant to additional future treatments. So this comes out with a statistic that is kind of frightening, that tumor recurrence after conventional treatment is highly likely. I think nearly 100% of glioblastomas recur, and significant percentages of ovarian cell cancers and breast cancers also occur. So what do we do about this? There are many fields, as we said, we're working very hard to treat cancer cells. And a very promising field that has come up recently is gene therapy. I'm excited to talk about this because this is what I was mainly the project on. But gene therapy is essentially introducing new genetic material into cancer cells so that you can manipulate the genetic material in them so they'll be more vulnerable to additional treatment. You can imagine if we send in a DNA plasmid into a cancer cell and it integrates with the DNA of the cell, we can cause it to express all sorts of things. And we can make it develop antigens so it can be targeted with antibodies, or we can just allow them to be more susceptible to radiotherapy or other drugs. In general, these gene therapy targets can be to silence oncogene or restore anti-oncogenes, or even stimulate the innate immune system of the body so they can actually recognize and attack these cells. Unfortunately, we're limited right now by the delivery method we can use to deliver gene therapies. There are a couple promising prospects. One is viral vectors. I think some of us may have heard about, where we send in a virus to target gene cells and to deliver DNA that way. And also, we have organic nanoparticles that we're starting to explore, where we send in nanoparticles to transfect cells. Unfortunately, there are a few limitations because this is a fairly new field that we're still trying to work towards. Viral vectors have a few safety concerns that we're trying to overcome, especially with mutagenesis, insertional mutagenesis, where inserting the DNA through viruses can actually cause different cancer growth. And organic nanoparticles, as you can see on the bottommost picture, is kind of just a fatty layer that surrounds any DNA or therapeutic you want to deliver. And we have very strict regimens on how nanoparticles size-wise can be. If they're less than 10 nanometers, then they'll be excreted by the kidneys very quickly before they can deliver. And if they're larger than 100 nanometers, then they get targeted for deletion inside of the cell, or targeted for degradation inside the cell, so they can't deliver anything either. Unfortunately, organic nanoparticles are a little too variable for us to consistently synthesize, so we need a new method to deliver these nanoparticles. And that's where my project kind of comes in. We are exploring the use of inorganic core nanoparticles. You can see on the picture here, there's a lot going on, but I think I would like to direct the focus mainly towards the inorganic core that's in yellow. This is usually metal, for example. In our lab, we use iron oxide. It's just a little metal bit that can use as a rigid body that you can add other functional moieties to. And this is very powerful because the more that you can add to a rigid base, the more function you can give to a nanoparticle. So for example, you can add some coating, for example, a polymeric coating that can allow it to attract and complex with DNA better. And also you can add targeting ligands so they can actually target the cell and deliver your therapeutic very precisely. We're going to focus on using the delivery system to deliver plasmid DNA, although there are other genetic materials that you can give. Okay, so I've kind of given a brief introduction, but what was the actual experimental design? We had just a few steps in our preliminary experiments. We first obviously had to synthesize the nanoparticles. We chose iron oxide cores to complex with, this is a scary word, XPEI and also CHSR10. These are essentially just polymers. So PEI is a polyethylenamine. It's just a fatty layer that is positively charged, and because DNA is negatively charged, it creates a nice attraction between the two things. And cholesterol is just another fatty polymer, and R10 is a targeting ligand, so we can actually penetrate cell membranes and target cancer cells very precisely. I've stated before that the therapeutic nucleic acid we used was DNA. Mix those all together, you add them to some cells. In our case, we used mouse breast cancer cells, 4T1 and MMC. And then we decided in our DNA to add a little component where it could express a specific fluorescent protein. So afterwards, to make sure our therapeutic worked, we just wanted to observe that it showed expression through fluorescence. So what are our results? Our results is kind of promising and very exciting. We saw that in synthesizing our organic or inorganic nanoparticles, that we were able to control the physicochemical properties very precisely. So the left-hand graph, your left-hand graph, shows the size constraints. And remember, I wanted our nanoparticles to be in a 10 to 100 nanometer range. And you can see that we were able to create complexes with and without DNA that were within that range. So that's promising. We can deliver our DNA within a very small size that can actually get to cells. And the other thing we had to consider is the zeta potential or charge of these complexes that we're making. Transfection generally occurs better when your overall particle is positively charged, because when they're absorbed by the cells, the positive charges can create a proton sponge, which when they're absorbed by an endosome, can cause basically the endosome to explode by uptaking more water, and then you can actually deliver your therapeutic more effectively. So that was a very exciting result. And the other thing we wanted to make sure is that our nanoparticles wouldn't degrade over time. So if we were to store them, that they would be stable. And we were able to see on the left side graph again that it's a fairly small time range, but 2 weeks, you can see that the nanoparticles exhibited negligible size fluctuations. This is good news for us because this means that we can consistently deliver therapeutics even over a great amount of time, and we can store them consistently. The graph on the right, or the figure on the right, I should say, is a gel electrophoresis gel. Kind of redundant, but normally if you have experience, you can kind of see that the top part where I have nanoparticle NP with DNA, those first three seem to have nothing in the gel, which would be concerning, but this is actually a promising result for us, because this means the way that gel electrophoresis works, you add a dye, and the dye intercalates with the DNA, and then it causes it to express very brightly. But if you complex DNA with something else, then the dye can't penetrate the DNA, and you don't see any expression. So it's actually good news for us that the DNA doesn't fluoresce, because this means that we were able to form very tight complexes with our DNA, and the fact that they didn't move down the gel means that they were large enough so that they wouldn't get through the pores of the matrix. So that's all very fine and well. Oh, okay, sorry. That's all fine and well. But how did they actually do in cells? So we were able to transfect mouse cells, and the fluorescence shows that we were able to do it very well and approximately equal to industry standard, very nice bright lights, and that they were less cytotoxic than industry standard transfection agents. Okay, so future plans is we want to be able to optimize our synthesis procedure a little bit better and improve transfection rates. It's a very promising field, and we've only explored a little bit of it, so I'm excited to see how this field develops in the future. I'd really like to thank Dr. Ellenbogen, Mrs. Ellenbogen, and all the mentors that have supported.